This morning I presented and meet the expert about single-cell approaches in myeloma, and I think this is really a very exciting technology that has been exploited so far mostly in the basic research, in the discovery, looking at new targets, and we've been hearing a lot about single-cell analysis also in characterizing myeloma cells and the tumor microenvironment also in this meeting. And I shared the data from our group. We work with the Weizmann Institute on this for a few years now already, and our main interest is actually into exploring mechanisms of drug resistance, and also being able to predict response and resistance to various combinations of drugs. So I described some data that we have published a year ago, where we were able to find a resistance signature to proteasome inhibitors in a trial that was focused on patients with functional resistance to their first-line therapy. And I also showed how this data allowed us to evaluate changes in pathways in these cells to do with the resistance, which genes were upregulated, which were downregulated, also to be able to look at the changes over time in longitudinal samples and actually track clonal evolution over time, and see what happens in those patients who undergo exchange of their sensitive clone with a more resistant clone, and how we can actually look at this with this tool of single-cell RNA sequencing, which is like, a molecular microscope, and we could actually see the pathways that are changing and tiding with this change. And finally, I proposed that perhaps in the future we will find a way to actually apply clinically single-cell RNA sequencing towards precision medicine, and towards being able to predict which is the best combination of drugs that would work for a given patient at a given time based on their transcriptional maps. Because right now I think this is one of the key challenges in the myeloma field, being able to select between really an expanding number of very potent drugs and immunotherapies, how to choose the best one, how to actually sequence them. So our vision is that this tool of single-cell RNA-seq would be able to contribute towards an answer to these important questions.